Welcome to Mr. Chalk's Revision Tips. In this video, we will look at the Lac Operon, repressors and promoters. So the Lac Operon consists of three structural genes and a promoter, terminator, regulator and operator. So these three structural genes are Lac Z, Lac Y and Lac A. Lac Z encodes for the enzyme beta-galactosidase. Lac Z encodes for an intracellular enzyme that enables more lactose to go and enter into bacterial cells. So an operator is a group of genes within a prokaryotic DNA that carry out a specific task within the bacterial cell. The genes to produce proteins which enable the bacteria to break down lactose only need to be activated when lactose is present, specifically when there is a lack of glucose for the bacterial cell to metabolize. The repressor protein is bound to Lac P and Lac O when there is no lactose present in the cell, which inhibits gene expression. So this is an example of negative control. An operator, like we just said, is a group of genes on a strand of prokaryotic DNA that carry out a specific task within bacteria. The genes to produce proteins to enable the bacteria to break down lactose only need to be activated when lactose is present. Proteins coded by the genes Lac Z, Lac Y and Lac A are required for the bacteria to break down lactose. Lac Z codes for the protein beta-galactosidase, which breaks lactose down into glucose and galactose. Lac Y codes for proteins lactose permease, which allow lactose to enter the bacterial cell. Lac A gene encodes for a transacylase enzyme. So the enzyme's cellular role may to be detoxify non-metabolized things by acetating them and preventing them to go and build up in the cell. RNA polymerase needs to move through the promoter region, so Lac P, and the operator region, Lac O, to get to these genes. So a repressor protein is bound to Lac P and Lac O when there is no lactose present within the cell, which inhibits gene expression. So this is an example of negative control. When lactose is present, it binds to the repressor and causes conformational change in the protein, causing it to break away from the operon. So to increase transcription, a cap protein also needs to bind to the opposite side of the gene. A high concentration of glucose inhibits the cap's ability to bind to the gene, as this will only operate in the presence of cyclic AMP. And cyclic AMP is produced when ATP is broken down into cyclic AMP and 2-phosphate by adenosine cyclase. So adenosine cyclase is inhibited by glucose. So when the cap protein is present and when the repressor protein is broken away, the RNA polymerase can move down the operon and go and transcribe those three genes into proteins required for the bacteria to go and metabolize lactose.
Thanks for watching.